The overall goal of this procedure is to perform in vitro uncoating of HIV-1 cores. This is accomplished by first producing HIV-1 particles. The virions are then concentrated by ultracentrifugation. The next step is to isolate cores by sucrose gradient ultracentrifugation. Finally, a kinetic assay of HIV-1 uncoating is performed. Ultimately, results show time-dependent uncoating of HIV-1 cores through quantification of Ca content in the pellet and supernatant by ELISA.
This method can help answer key questions in the HIV-1 field, such as the role of cellular factors in uncoating and the effect of mutations on virus capsid stability. Before proceeding, you must obtain permission and follow guidelines from the Biosafety Office of your institution to work in a biosafety facility for infectious HIV-1. Proper handling and care should be taken while working in a biosafety laboratory.
In this demonstration, HIV-1 particles are produced by transient transfection of 293 T cells with HIV-1 proviral DNA using a calcium phosphate transfection method. The day before transfection, obtain previously cultured 293 T cells and detach them from the plates using 0.5% trypsin EDTA. Seed 6 100mm culture dishes for production of virions. The following day, the cell monolayers should be about 25% complement and ready for transfection. For 6 dishes of cells to be transfected, use 120 micrograms of proviral DNA to prepare a mixture containing calcium chloride and best buffered saline as described.